This video demonstrates step-by-step procedures for deriving a virtual genome using virtual tissue technology, CellSim, developed by Crowley-Davis Research. Specifically designed for studies in cancer biology, drug discovery research, bioinformatics, or in silico toxicology. CellSim V tissues are based on a virtual genome informed by bioinformatic or patient-specific data. Both complement and guide wet bench research by identifying gaps in the knowledge framework and enable experiments that are impossible to perform in vivo. Evolutionary Search allows researchers to derive potential virtual genomes that provide valuable insights regarding disease mechanisms and their early warning signs. A virtual genome is defined as the genetic information, properties, and algorithms needed to support the development of a desired V tissue or V-organoid from one or a few starting cells. Working backwards from the properties of a tissue to the set of inherited components and processes needed to form and maintain such a tissue can potentially transform research in cancer biology and related fields. Why and how? Researchers often know a great deal about the diseased tissue, but understand less about the precise inner workings of how the disease arose. As explained below, deriving a potential genome can provide valuable insight regarding disease processes and their early warning signs. Following a survival of the fittest strategy, CDR developed an advanced evolutionary search process to identify a virtual genome that supports the development of a desired V tissue, starting from one or a few starting cells. As indicated by the red question mark, our main challenge is this: starting with patient-specific or bioinformatic data and basic biochemical and physiological properties of a particular tissue, can we determine, via evolutionary search, the genetic makeup that gives rise to such a tissue? Essentially, this is development in reverse, starting with the tissue, then exploring plausible mechanisms for building the tissue. This powerful evolutionary search process rests on iterative refinements of an initial candidate virtual genome over many successive cycles of steps as follows. 1. Create slight variations of the initial prototype candidate genome using genetic mutations. 2. Grow virtual tissues from each of these varied genomes, thereby creating a collection of possible V tissues that comprise one generation. 3. Compare each of the resulting V tissues to the target tissue. 4. Select the genome whose V tissue most closely resembles the desired tissue, those with the highest fitness index, and use their traits to populate the next generation. Repeat the above steps until a genome that supports, meets, or exceeds a predetermined degree of fitness has been found. In the remainder of this presentation, we're going to focus on a simple V-organoid, shown here in the lower right, and as seen in CellSim's client phenotype viewer. It forms a compact ball of approximately 22 cells in a state of homeostasis or relative stability. The V-organoid can be described or represented in several ways. The most complete description is in a configuration file that includes the virtual genes, initial chemistry, metabolic processes, and their interactions, as well as supporting algorithms. This is written in a structured file format known as XML, or Extensible Markup Language. And it's a format that's both human readable and machine readable. Later we'll examine XML files in detail. In the lower left-hand part of this slide, under B1, is the cellular process summary that indicates how this V-organoid develops from a single cell and achieves homeostasis. In this example, growth and division are driven by nutrients. Cells on the inside of the growing cluster are protected from death by contact with their neighbors. As the cluster gets bigger, cells near the surface may die because they become stressed and lack such protection. These two opposing tendencies, cell growth and division versus death, are shown in the pseudocolor images onto the lower right-hand corner of the screen under B2. As a third option, interactions between components are represented as a network diagram known as a gene and metabolic regulatory network, or a GMRN. In this depiction, the internal virtual biological machinery is represented in a gene and metabolic regulatory network diagram. The parameters and coefficients in this diagram control the inner workings of each cell's metabolism in a complex emergent sequence of steps that produces the V-organoid phenotype via the ontogeny engine, as was shown earlier in slide 2. These parameters are also subject to mutation during evolutionary search, as we will see in a moment. Now that we have a basic understanding of our V-organoid, let's start examining the how-to operations involved in an evolutionary search. To begin, we need to do two things. Define a desired endpoint, V tissue or V-organoid, and select a prototype virtual cell from that V tissue. This cell will then be used to seed and propagate a population of similar candidate virtual cells, each of which may develop into some type of V-organoid. Each of these V-organoids is compared to the desired result and assigned a numerical fitness index between 0.0 and 1.0. In this following example, we'll see the fitness of simple V-organoids. Basically, three phenotypes grown from differently mutated cells derived from the prototype virtual cell. In the left panel, the phenotype grown by the highest ranking virtual cell is shown with a fitness value of 0.997, virtually 100%. The other two phenotypes were grown by cells which contain different mutations. The resulting virtual cells may develop different phenotypes because of introduced mutations which alter development and lead to lower fitness. Now let's walk through the procedures involved in evolutionary search using the user interface control and drop-down menus in CellSim. So we're going to accomplish two things in this first part of the evolutionary search demonstration. One, we'll define a target V tissue. And two, we'll select a prototype virtual cell from that V tissue. This prototype virtual cell will then be used to propagate a population of similar candidate virtual cells. Each of the candidates is grown into some form of a V tissue, which is then compared to the target V tissue using a complex set of metrics grouped across a wide range of different domains from cell adhesion to cell signaling. The highest ranking virtual cells from one generation are selected to populate a subsequent generation, and the cycle is repeated for as many generations as are needed until an individual virtual cell is capable of growing a V tissue within a predetermined fitness metric. We'll start with an elementary V tissue known as a self-organizing cluster, shown growing here in CellSim's client phenotype viewer. It consists of a group of cells that continue to grow in a state of homeostasis or internal stability. Such a V tissue is alive in a virtual sense as long as the power is on and the ontogeny engine is operational. As in its counterpart's existence in vivo or in vitro, V tissues respond to real-time perturbations in much the same way as do living systems. To begin, we'll pause a stable V tissue as it has advanced approximately 1,000 steps. We'll also save the V tissue as the target for our evolutionary search by simply using CellSim's checkpoint feature, which stores the complete ontogeny of the V tissue as follows. We'll pause. We're now at 952 steps. We have 28 cells. We'll now checkpoint the V tissue as it stands, which includes its entire ontology. That is, every biological detail is being preserved in this checkpoint feature. We simply click this and we'll put a file name on it. We'll call this test 1. Continue. Next, an arbitrary cell is selected and will be used as the prototype virtual cell that will propagate a population of slightly mutated cells. We'll pick this cell here arbitrarily, and we'll note its cell ID number because we'll need that to get its network diagram. That's 24401. And we can demonstrate the network feature by simply going to our static metabolic network screen. We have a list of all the cells, the cell ID numbers comprising the V tissue. All of these cells are shown, but there's only one that we're interested in. That happens to be this particular number. So we'll go to 24401, select that, and we'll get our diagram for that particular cell as shown here. We can change the view just slightly so we can see the entire affair. And we see that we have our correct cell number. We're at 952 steps, which corresponds to this here. However, the most important representation of the cell's characteristics is a structured file format known as XML, which stands for Extensible Markup Language. This is a format that's both human readable and machine readable. That particular file is here. As you can see, it's a little cryptic, but it can be read. You can understand it and actually make changes to that file. But more importantly, it fits a machine format for automated research, which is what our evolutionary search accomplishes. We'll save this particular file as such, and we can do that by simply let's cancel that. Okay, we'll make this test1.xml and we'll save. Now that we selected an appropriate prototype individual, we simply save it as we just did, and that along with our checkpoint save satisfies our two objectives. We have both the target virtual tissue and we have a prototype virtual cell. Next, we'll cover the Evolutionary Search client, a configuration framework in which various aspects related to the field of genetic algorithms, or simply GA, can be established and adjusted to suit a particular project or series of experiments. Note that a configuration-wide save and load option is available for storing the entire project settings. As the GA client screen shows, there are 6 configurable groups of controls that allow an evolutionary search to be conducted efficiently. Since this is an overview, we won't go into great detail concerning each configuration group, but point out by actual example how the process functions. We'll start with conditions, which specifies the desired fitness and maximum generations for an experiment. Next we have an initial population. This specifies the population size and the number of mutations applied to a population. Fitness specifies the ontogeny engine to use, search criteria, and a reduction strategy, if any. Replacement specifies the method used to repopulate successive generations of candidates' virtual cells. Mutation allows a delineated segment of candidate virtual cells' genomes to be potentially mutated. And selection specifies a method of selecting the best candidates from a given generation. Additionally, each group has its own import-export options, allowing users to save or retrieve a particular group's settings. We'll now explain each of these configuration groups in more detail. The conditions group is relatively simple, as it consists of only the acceptance metric in terms of the fitness of a candidate's virtual cells V-organoid compared to a predetermined standard. Likewise, the number of generations is specified as a maximum, after which the entire run will terminate if the target fitness has not been achieved. The initial population group settings center around the prototype virtual cell covered earlier in this video. In addition to the usual settings import-export feature, there is also an edit option as well. Note the term individual refers to our prototype virtual cell specifically in this example, whereas in a broader context it refers to any such construct that may serve as a potential solution to a given problem. Here we could specify the number of initial mutations to apply to a population. In this case, 15 mutations will be applied randomly across the entire population of 120 as entered in the multiplier box. The individual's custom name, test1.xml, is relevant as the mutation settings key off that name when mutations are applied. The fitness group settings are considerably more involved than the previous two groups, since this allows a range of options that can be tailored for narrowly defined evolutionary search projects. In order to keep this demonstration somewhat straightforward, yet relevant to the actual experiment at hand, we'll specify the endpoint V-tissue or organoid we saved earlier as a snapshot file. Note that this will be not only the complete array of all the virtual cells that make up the V-tissue, but their individual cells' constituent elements as well. For example, genes, molecules, resources, metabolic relationships, signaling pathways, and so on. The goal of correctly configuring the fitness group of settings is to enable a comprehensive set of disparate metrics to be aggregated across different domains such as physics, electrical, chemical, and biological into a single unit of measure that reflects the entire V-tissue composition. Replacement is the last step in the breeding process of the GA cycle. The technique chosen is used to decide which individuals stay in a population and which are replaced on a par with selection in influencing convergence toward a solution. We have available three of the more popular methods. Tournament selection, which involves running several tournaments among a few individuals chosen at random from the general population. The winner of each tournament, the one with the best fitness, is selected for crossover. Generational selection, where the entire population gets replaced each generation through the processes of selection and reproduction. And most fit, where the fittest individuals are selected for reproduction in order to produce offspring for the next generation. We use this last method in our demonstration. The mutation group can be, and usually is, the most elaborate configuration settings in an evolutionary search procedure. It involves many detailed options scaled along the prototype virtual cell's biological makeup within the cell. Like the fitness group settings discussed earlier, mutation operators can be activated along one or more biologically specific lines, and may also include domains spanning physics, for example, plasticity, rigidity, etc., as well as chemistry, genes, molecules, resources, metabolic relationships, signaling pathways, and so on. The selection shown under the Example Mutation tab gives a glimpse of some of the biological choices where mutation can be turned on. The selection operator group determines the fittest individuals and lets them pass their relatively superior attributes on to the next generation. Maintaining good diversity in the population is extremely crucial for the success of a GA. Taking up the entire population by one extremely fit solution is known as premature convergence and is an undesirable condition in any GA. In the roulette selection, a circular wheel is divided into n number of pie-shaped pieces according to fitness. A fixed point is chosen on the wheel circumference and the wheel is rotated. The region of the wheel which comes in front of the fixed point is chosen as the parent. For the second parent, the same process is repeated. The all selection is simply a normalization of all fitness values. Normalization means dividing the fitness value of each individual by the sum of all fitness values, so that the sum of all resulting fitness values equals 1. The number of ensuing offsprings is in direct proportion to a parent's fitness rank. The random selection process proportionately mates top-ranking individuals with randomly selected individuals from the lowest ranking portion of the population, until the next generation's population size is reached. After we've completed our 6-step configuration process, we simply start an experiment by clicking the Create Experiment tab in the upper left-hand part of the GA control screen. Then click Run at the bottom of the screen. Depending upon the nature and complexity of the search subject, hardware speed, and adequacy of bioinformatic detail, typical experiments can take a few hours to several days to reach a satisfactory conclusion. All results are shown in descending ranked order, both throughout and at the end of the experiment. Results can be saved in their entirety to a desired location. This concludes the Evolutionary Search demonstration video. Thank you for watching. Links to other CellSim resources and videos are available in the description below and in these links of the final slide.